Yes, little children, ready to score 30 on 30 in your practical board examination for biology? Well, I hope you say so after watching the whole video. So I have put in a lot of effort to make this video. So I hope you follow it properly, and if you're someone who has no idea about practical examination, this will definitely help you well, because I am going to explain everything in as much detail as I can. Okay, so let's make it short and sweet and also very much helpful. So let's get into it, my dear children. Why is it not moving? Yes. So the practical exam is out of 30 marks, right? And how do you divide the marks? Basically, the experiment and the theory that you're going to do is going to give you 20 marks. Okay, the record that you're going to maintain will be giving you 6 marks. The viva that you're going to appear will be giving you 4 marks. Together, you'll get 30 marks. So, are you going to score 30 marks? Yes, we are going to score 30 marks. How? Let's see. So, basically, this is how the question paper looks. The first question which is there, it has 4 experiments. Ma'am, how can we do the 4 experiments? You don't have to do all the 4 experiments. You just have to do one. So, basically, sometimes in some schools, it's dependent on the lottery system where everyone was given a chit to pick, and you can have the own experiment. So, you will get any one of these experiment, or in some school, probably some children might be said that you have to do experiment number 1, that is this, and then you have to do experiment 2, and things like that. So, anyway, you have to be prepared for all the 4 experiments, because you could get any of this. Now, the first one is a pollen germination experiment. The second one is pollen tube growth experiment. The third one is transverse section of ovary, where you have to cut the ovary and see the number of locules and placentation. And the fourth one is basically you have to see the cells of the plant. Okay, the plant cells, observe it under the microscope and see how many nuclei are there. Now, you might be worried that, what are you saying? I'm not able to understand. It's totally fine. It happens. So, do not worry. Okay, I am going to talk about each of them separately as well. So, any of these things, if you do properly, how many marks do you get, my dear children? You get 5 marks. The distribution also, I will be talking about that. Now, this is something that you have an option, right? Okay, anyone can get any experiment, but there is an experiment common to all, and that is given in the second question. So, you have to make a temporary slide for showing the stage of mitosis, my dear children, which is done in onion root tip. So, that is common to all anyway, no one can skip it. You will get 5 marks for this particular question. Now, the third question is basically, you might be given 5 pedigree chart, you have to identify one and comment on that. Gives you 3 marks. Now, again an identification question, again 3 questions over here is again identification. So, in total you see 1, 2, 3, 4. 4 of them you have to identify and then comment on it. So, you should be having enough knowledge about it, then only you will be able to identify it. I am not telling to scare you up, but I am just giving you a heads up that yes children, you have to be prepared. Okay. So, I cannot be just sweet over here and tell that, hey, everything it will be done, you don't need to do anything. It's not like that. We have to do our part. Right. So, yes, now we are going to talk about each question in detail. Question number 1, experiments. There are 4 experiments. I am going to talk about all of them. Okay, basic thing, not in very much detail, okay, otherwise this video will go too long. So, pollen germination experiment, there you will be getting vinca rose, which is a flower, okay, which is a pink color flower that you are seeing over here in the background. And what you have to do, you have to take a glass slide, you will be having, you will be given sucrose solution, my dear children. You have to put the sucrose solution on the glass slide, and then you have to dust the pollen grains of this particular flower. You have to dust the pollen grains onto the slide that you are having, and then you have to put the cover slip and put it under observation for some time. Okay, let's say 15 to 20 minutes it will take. Why are you waiting? Because in the presence of the sucrose which is there, the pollen grains which is there, it is going to germinate. It is going to form what, my dear children? It is going to form a tube. Okay, pollen grain is going to form a tube over here. So, I hope you know that the pollen grain has plasma membrane, and then it has one more covering which you call as intine, and then it has the outermost covering which is made up of a tough resistant substance which is known as, and this particular layer is known as exine, and the resistant substance is known as poropollenin. So, somewhere there are area where the sporopollen is not there. So, there are like pores over here. What do you call these pores? You call these pores as germ pores, my dear children. And I hope you know that inside that you have what? One generative cell and one vegetative cell. Okay, you will be having one generative and one vegetative cell depending on the type of plant it is there. Okay, but anyways, what happens is that when the pollen grain lands on the stigma, usually, slowly the intine thing that you see over here, okay, the intine thing that you see over here, slowly will be making a tube like this. This is known as pollen tube with germination, my dear children. And it goes through the germ force where the sporopollen is not there. You get it? And later what will happen? This guy which is there, okay, the generative, generative and vegetative cell is there. The generative cell will divide itself, make two male gametes, and the male gametes will finally move down to meet the female gamete which is there in the ovule. So this is how things work. Okay, so in the presence of sucrose, they are able to do germination, which is known as basically the pollen tube growth will definitely happen. Okay, now what you need to do is you have to prepare the slide. If you prepare the slide properly, you get 3 marks. Then you have to make a table for calculation, which will give you 1 mark. If you are not going to prepare this table, you are going to lose a lot of marks. So children, when you put the pollen grains on the sucrose solution, and now you are seeing it, obviously under the microscope, you will be having a view like this. Now you can divide into 3 regions. Suppose this is a region, this is second region, and this is third region. This is region number 1, 2 and 3. So what do? You observe the number of pollen grains over here. So 1, 2, 3, 4, 5, 6 pollen grains are over here. 1, 2, 3, 4 pollen grains are over here, and 2 pollen grains are over here. So what do? Total number of pollen grains in 1, 2, 3. What is this 1, 2, 3? These are the segments that you are having. So in 1, you have almost 6. So you will be writing over there 6. Cool. Now, how many of them, the number of pollen grains germinated, how many of them are germinating? Let's see. So germination means they will be forming a tube-like structure like this. So I see 1, 2, 3 of them are germinating. So I have to write like that 3 of them are germinating over here. Similarly, the same goes with the second one and the third one. In total, if you do, the totaling of it, if you do, you get the value of capital N. You get the value of capital N. Similarly, over here also, you will get the value of small n, my dear children. Now, there's a formula over here, as you can see, n into 100 upon capital N. You have to replace these numbers over here and you will get the answer. So, this formula is very much important. This formula itself is really important. You will get half marks for this. Now, when you calculate it using this particular formula, whatever answer you get, this will also give you half marks. So, half and half, 1 mark. Okay. Table gives you 1 mark. The preparation of slide gives you 3 marks. In total, how many marks? 5 marks. You got it. You got it. Okay. Suppose, okay, the germination is not happening. It's okay. You still have to put 0 over here. Okay, you still have to put 0 over. None of the pollen grains germinated. 0, put 0 and explain it to them. That is what they want. Put the formula over here. Substitute it with the things that you are having. Okay, anyways, this won't be empty. You would be having something over here. So you replace the capital N with this. You put 0 over here. The answer will be 0, but it's okay. You have to show it. So calculation of pollen germination goes like this. So I hope this experiment is clear, my dear children. Please keep it for 15 to 20 minutes. And by that time, when you are waiting for it to germinate, do not waste your time. Please write whatever is needed. Cool? Chalo. Next thing. Okay. So this is how it will look. Okay, the pollen germination and all, not exactly very clear like this, but yeah. Okay, this is how the pollen germination thing works. Just for you to understand. Now, pollen tube growth. Pollen tube growth is the next experiment that we have. Okay, again you will be getting the Vinca rose flower. So, you know this structure, what do you call this structure? Do you know what do you call this structure? Basically it's a pedicel. Okay, it's your pedicel. Anyways, so what you have to do is you have to dissect this. You have to cut this open with the help of the needle and things like that. And then you will be able to observe what? The stigma and style over here, my dear children. You will be able to observe the stigma and the style over here and also the ovary, but it won't be very much visible. When you pluck it out, the stigma and style will be coming out. So it will appear to you something like this, a very small structure that you get. Okay, very small structure that you get. Now, what you need to do is that when you put it on the slide, okay, when you put it on the slide, you make sure it is not dehydrated. You have to put water and things like that. Okay, and then what you have to do is you put the cover slip. Okay, you're going to put the cover slip, and before putting the cover slip, you have to stain it as well. Okay, wait. So first thing that you are going to do is you are going to take the stigma and the style. You're going to treat it with some hot water. Okay, you're going to treat it with hot water, or probably you're putting inside a watch glass that is having water and probably heat it up. Which, why are you heating it? Because you want to make sure that the cell wall and everything is now going to get broken down, such that the stain that you're going to put can be taken up easily by the plant cells. That is why you're going to treat it with hot water first. Okay, now over here you are going to take a stain which is known as cotton blue, my dear children. You are going to take cotton blue and you are going to put it on the slide where this particular thing is there. Okay, the stigma and the style that you have collected, you are going to put it back onto the glass slide and you are going to put cotton blue, and then you are going to cover it up. You are going to cover it up with the cover slip. You are going to remove all the extra stains. Please make sure there is no air bubbles as well. You have to make sure there is no air bubbles. Fine. Now, that is about the preparation of the slide. Now, you have to see it. When you see it, probably you might see something like this. Okay, something like this. What is this? This is the pollen tube that is forming. Even if it is not visible, it's fine. You have to draw this diagram and you have to make sure that you are labeling pollen grain, you are labeling stigma, you are labeling pollen tube, and you are labeling style. 4 things. Pollen grains gives rise to pollen tube, stigma, below it what do you have? Style. These 4 things are really important. If you're not making the diagram, you are going to lose 2 marks. Half marks each. So half, giving you 2 marks. 2 plus 3, my dear children, 5 marks. Okay? Fine? Cool? So these things you have to be sure. Cotton blue is the stain that you are going to use up here. So the steps and all you just go through, because in every other books you have lot of other things. So which is why I do not want to get you confused over that. Just go through the procedure by your own self. I am going to tell you here that what you should not be missing out for your marks. Cool? So yes, and also a little bit of reasons also. So whenever you are heating the cells, the plant cell and all, you are making sure that the plant cell is able to take up the stain that you are going to treat it with. That is why you are heating it or probably boiling it in the boiling water. Okay? Cool? Chalo. Next thing. Transverse section of ovary. Again, what do you have to do? You have to take hibiscus plant. Okay, you will be given hibiscus plant, my dear children. You have to cut it with a needle. This thing that you have to cut it with a needle, and when you open it, you will find some structure like this. So there is ovary up here. There is ovary up here, and then you have your style, my dear children, and over here you have the anther filament, and at the tip you will be having what? Basically the stigma as well. Okay, you will be having what? The stigma as well. And over here, let me tell you, the stigma which is there. Okay, so you will be seeing there are multiple carpels. Okay, there is stigma, so there are so many stigmas up here. So what do you mean by so many stigmas? Probably there are multiple what? Carpels. What do you mean by carpels? The female reproductive system. Okay, the female reproductive system. So usually it looks like this. Okay, you have the stigma, the style, and there is one ovary. Now suppose there are multiple carpels, you call this condition as multicarpellary condition. You have so many carpels. Now when these carpels are fused together, okay, when they are fused together, you call this condition as syncarpous condition. Syncarpous, all the carpels are fused together, and this is known as syncarpous condition, my dear children. Okay, that is what exactly is happening over here in the case of hibiscus. Now going ahead, so over here, you have to prepare the slide. How do you prepare the slide, my dear children? Again, you are going to take the ovule, okay, sorry, the ovary that you are having, and the ovary you are going to cut it like this. Okay, the ovary that is there, you are going to cut it like this, and you are going to put it in the glass slide that you are having. Okay, in the glass slide, you are going to cut it and you are going to put it, make sure there is some water in there, so that this does not get dehydrated. Otherwise, you won't be able to see it properly. Okay, and one thing that I missed over here in the previous one was when you are putting it, okay, when you are putting it after staining it, no, you have to make sure you have to press it. You have to press it, then only you can see it. Okay, without pressing, you won't be able to see. You can press with the back of the pencil or things like that. Okay, also with your hand also, but I won't suggest doing with your hand. Do it with the tip of the pencil. That will be much better. So over here, what you did, you took the ovary. Okay, you took the ovary, you cut it like this. This is the transverse section. Okay, when you cut it, you will be finding something like this. And you are going to observe it under the microscope. You will be finding one, two, three, four, five. 5 locules will be there. 5 locules will be there, my dear children, and 5 locules means there will be what? Inside the locules there will be ovule also. You will be having multiple ovules, my dear children. Okay, there will be, sorry, I was going to fall down. So there will be ovules also, but how many locules you have? You have 5. So what do you call this condition? Pentalocular condition. Pentalocular condition. Now this thing you have to answer. Report the number of locules in the ovary. There will be 5. So you call this pentalocular. This is 1, this is 2nd, this is 3rd, this is 4th, and this one is what? 5th, my dear children. Okay, now going ahead, these are what? The ovules which are there in the ovary. Now talking about the placentation which it is having. So the placentation, as you see over here, okay, I'll talk about one extra thing. So if you see the green piece, the ovules are arranged like this. So it is marginal placentation. What do you call this? Marginal placentation. But over here, okay, because all of them are on one particular slide. Over here, if you see, so it looks something like this, where there's little amount of placentation in the middle, and there are ridges separating it. Okay, so what do you call this condition as? You call this as axial placentation, or not, yeah, placentation. You call it as axial placentation or axial placentation. Either way, you can say it. Okay, so actual picture, it will look something like this. Okay, not exactly like this, obviously. But yes, this is what I was saying. So there will be a little amount of placenta separated by ridges that you can see over here, making 1, 2, 3, 4, 5 locules, and in between you have so many ovules, my dear children. What do you call this placentation as? Axial placentation or axial placentation, either way. Okay, but you have to write it down. For writing it, you get 1 marks. For writing the number of locules, you get 1 mark. So pentalocular condition, okay, pentalocular condition, and also axial placentation. I'll write it over here. Pentalocular condition and axial or axial placentation. Just give me a second, my dear children. Okay. So yes, again, you will be getting 5 marks for this particular experiment. Done? Chalo. Next experiment. Yeah. Next experiment, preparation of nucleic acid staining experiment. So this is a very easy experiment, okay. So I hope you know that onion peel if you take, okay. If I had an onion over here right now, I would have explained you by showing it. So this is the outer area, this is the inner area. The inner area, if you see, if you can peel it out, okay, you can slowly peel it out, there will be a very thin membranous thing coming out. Okay, this is a peel that comes out. The inner peel you have to take. Okay, you have to take the inner peel, my dear children. Cool. And then what do you have to do? This particular inner peel, again, you have to put some water on it. Okay, or probably you can take a watch glass, you can put some water and you can put this peel and you can heat it a bit. Why you are heating it? Because probably you are going to add some stain into it so that it can take up the stain. So when you are heating it, so probably you are making sure the structure of the cell wall is disoriented such that the stain can be easily taken up. And also for the nucleic acid, the nucleic acid which is there in the plant cell, in their nucleus, it can also receive that stain. Okay, so which is why you are boiling it. Now what you are doing? You are going to take another glass slide. Okay, you are going to transfer this particular glass peel into the glass slide. Okay, you're going to transfer this particular glass peel, this particular onion peel into this particular slide, and you're going to put acetocarmine. Okay, that is a stain. Acetocarmine stain, you are going to put it onto this. You're going to put a cover slip in 45 degrees angle, and then make sure there is no air bubbles, and then you can see it under low magnification power. Okay, every time whenever you do the experiment, start with low magnification power, 10x. Okay, that should be the first priority. Again, keep it like, so the microscope tip that you see, okay, and also the specimen that you have over here, it should be having almost around 1 centimeter gap. It should be having almost around 1 centimeter gap. Do not go touch it. Okay, please do not go touch it. Okay, whenever you need, you can go a bit lower. That is fine. Do the coarse adjustment and do the fine adjustment. There is coarse adjustment which is bigger. Okay, if you see the microscope, there is a bigger knob and a smaller knob. If you rotate the smaller knob, that will do the fine adjustment. You might be able to see it clearly. So don't go and make that thing go hit the specimen and get ink on it. Otherwise, you won't be able to see it, which is why I am telling you. First, use the lower power magnification, which is what? 10x, and then only go ahead with the next, but you don't have to go ahead with the next. You will be able to see it in the 10x itself. Cool? Works? No, yes. So that is what I was saying. Now, acetocarmine you are using for staining the nucleic acid, my dear children. Okay, it will stain really well the nucleic acids. Why? Nucleic acids, as in what? I am talking about the genetic material, that is DNA, RNA, whatever is there. So the DNA which is there, I hope you know, it is negatively charged. It is what? Negatively charged thing, my dear children. And this particular stain will, this acetocarmine will stain really well. This acetocarmine will stain the DNA really well because the DNA is negatively charged, whereas acetocarmine, which is a dye, has positively charged content. So positive, negative things attracts. Yeah, so it will be able to see it really, really clearly. So you will be seeing something like this. Now what, the preparation of slide goes like this, but what you have to report the shape of the cell. The shape of the cell, as you can see, they are elongated, and can I say, even though it's not properly rectangular, but I have to say that it is what? Rectangular. Rectangular. For this you will get 1 mark. Now reporting the number of nuclei, if you see all the cells over here has just 1 nuclei. This nuclei which is there, it is just 1, my dear children. So I say it is unicellular or uninucleate. Can I say it is uninucleate, as in it just has 1 cell, 1 nucleus, my dear children. Okay, which is stained very much properly, very much brightly it is being stained as well. Now one more thing, if you do not know, I'll tell you, the nucleus if you see in all of them, it is towards the periphery, towards the sides. So I'll say it is a peripheral nucleus. Peripheral nucleus. This is just for your reference, okay? It is uninucleate, that means there is only 1 nucleus and the cells are what? Rectangular, elongated rectangular cells. Cool? For this you get 2 marks, for this you get 3 marks, total 5 marks. Simple. So take the inner peel properly, heat it in the water, okay? Don't directly go heat it. Okay, put some water in the watch glass, put this glass peel, go heat it, take the glass peel out of it, put it on the glass slide, put some acetocarmine and then cover it up and then watch it. Now again, as I said, your teacher might have said differently, so go according to them because they are the ones doing the practical. I'm just here to tell you what you should not be missing out. Okay?
Okay.
Now, question number 2. So question number 1, 4 experiments are done. Now we are left with question number 2 experiment. There is only one which you all have to do. That is, make a temporary mount of the onion root tip. So children, the onion root tip will be provided to you. Okay, the onion root tip is provided to you. Usually the onion root tip is treated with HCl first. Okay, what did they do? Usually you treat it with HCl first. They might have already treated it, so you have to check it. Okay, so anyways, however your teacher taught you. So why do you use HCl again? To make sure that the cell is going to take up all the stain and everything really well. Okay, that is why you treat it with HCl. Now after treating it with HCl, you are going to take the root tips out. Okay, we are going to cut them into smaller pieces. Okay, now you're going to take it out, the tips, and then you're going to treat it with acetocarmine again. Okay, you're going to put acetocarmine on it, little drop. Okay, and then what? You're going to heat it. Okay, so that it becomes fixed. So you heat it a bit, this acetocarmine stain, you heat it a bit. Okay, and then what do? Basically you put glacial acetic acid. Now that is to fix the structure and everything. You put glacial acetic acid and you cover it up with the cover slip. Now these things go according to your teacher, okay, not according to me over here. These are the things that is given in the PU2 manual. Your teacher might be following something different, so yes. Okay, that's why I'm not stressing too much on the preparation of the slide, but I'm just giving you a brief idea of how things work. So acetocarmine is for staining the cells and the nucleus pretty well, because you are going to see about mitosis, right? And mitosis, basically you are going to see about the cell's nucleus, what is happening in there. Okay, the nuclear membrane will disintegrate, the nuclear membrane will seen, lots of things are going to happen over there. So for seeing it properly, you use acetocarmine, my dear children, and carmine. And why do you use HCl? So that you make sure that the cells are able to take up this particular stain. Okay, the structure is changed a bit. Okay, it is a bit more easy for the cells to take up the stain. For that you treat it with HCl, and now at the end you are going to treat it with glacial acetic acid. Why, my dear children? To fix the structure. Now you don't want any change. Now you don't want any other, things to get changed over there. Otherwise, the whole picture will be different, right? So which is why for fixing the structure of the cell, you are treating it with glacial acetic acid, and then now you are going to see it. Now you have to identify any one of the stage, my dear children. How many stages are there in mitosis? In mitosis, you have prophase, you have metaphase, you have anaphase, and you have telophase. This will look something like this. Okay, not exactly, but yes. So in prophase, what happens is that the chromatin fibers which are there, it is going to condense. It is going to condense to form the chromosomes. That is happening in the prophase. And also the nucleus and the nuclear membrane which is there, it is going to disintegrate. Nuclear membrane which is going to be there, it is going to disintegrate. So you don't have a proper structure of the nucleus, but it will stain something like this, like a ball of cells you would be seeing. Inside one cell, you will be seeing one particular whole bunch of stainy like thing. So here what is happening, the chromatin is getting condensed to form chromosome. The thin fiber like structure are going to form chromosome, and also the nuclear membrane which is there, it is going to disintegrate. So you have to comment also. So how you have to comment on any two stages. You have to identify one stage, my dear children. You have to comment on any two stages that is there. Cool? Identify one stage and comment on any of the stages. Now over here, metaphase that you are having, in that the chromosomes which is there, it is going to align in the middle. This is the most easiest thing to see. They are going to align in the middle of the cell. This is known as metaphase. Okay, they are going to align in the equatorial plane. Fine. And each chromosome you will be finding two chromatids are there, and it is attached to the centromere. This is the thing. So again I am going to make one over here. So these things are arranged in the equatorial plane, my dear children. Each chromosome have two chromatids. This is one chromatid that you see over here, and this is the second chromatid. Both of them attached to the centromere, and this is in the middle of the cell, which is why it is known as metaphase. Now anaphase is when the chromatids which are there, they are going to separate out. They are going to separate out, so it will appear something like this. So chromatid fibers, they separate, because the spindle fiber formation happens. The spindle fiber formation happens, which is why the chromatids which are there, they are going to separate. The spindle fibers, they move towards the opposite poles. Use this term. They move towards the opposite pole. The spindle fibers makes the chromosomes move toward the opposite poles. Hence, the chromatids will be separated. Fine. Now, again, over here, what do you need to do? Telophase. This is the last one. Telophase is where the cells are also, over here you see, this is getting separated totally, this is also getting separated totally, and slowly there will be formation of nuclear membrane around. Now this won't be very much clear, but I think it will be distinct from prophase. How? Because here there was only one bunch, but here you will be having two bunches at the total opposite end. Okay, so telophase is when they have completely separated, and now the nuclear membrane formation and everything takes place again. Okay, so you have to go according to that. So yes, that's all. So this will be giving you 1 mark, commenting on it will be giving you 1 mark, and identifying the stage will be giving you 1 mark, and preparation of the slide will be giving you 3 marks. Cool? So that's all. I don't think it's identification of one stage. I have to just take it. Just give me a second, okay? Just a second. So yes, a little correction over here. That is what I was thinking. There's something wrong. Yeah. So it is not any 2 stages. You have to mention any 2 points. So you identified one stage, and then you basically comment on it. Any 2 points should be there. Each point will give you half marks. Half marks each. Fine. Cool. So that's how things works over here. So be sure you know about them. Read at least 2 points for each of them. If you see the diagram carefully, you will be able to understand the points also. Chalo, this is done by all. This should be very much clear to you. Okay, don't mess around this. And the Viva questions will be coming. Why you are using acetocarmine? Why you are using HCl? Why are you using glacial acetic acid? And things like that. Okay, so be sure. And over here also, one more thing that I want to add up is in the in this particular thing, when you are going to cut the ovary, okay, the TS of the ovary, when you are going to observe it, right? So make sure, my dear children, that you are cutting in very thin slices. Not very thin, but yeah, thin slices should be there. If it is thick, you are going to lose your marks up there. So in preparation of the slide, if you are going to show them a slide which has very thick one, you will get 2 marks here. Okay, with very thick one you will get 2 marks. If it is thin, then only you get 3 marks. So you will lose 1 mark over there. So make sure you are cutting it in a thin manner. Cool. So yes. Chalo. Next one. Question number 3. So pedigree chart is there. So you have to identify any one of the pedigree chart given below. So it can be autosomal linked dominant trait, autosomal recessive trait, X-linked dominant, X-linked recessive, Y chromosome linked trait, and then you have to, any of this, after you identifying it, you are going to comment 2 points. Okay, you are going to comment 2 points. For each point you will get 1 mark. Okay, for each point you will get 1 mark. Over here for identifying also you will get 1 mark. Sorry, 1 mark. So here 1 mark, here 2 marks. What is the total marks? 3 marks. Okay, so how do you identify? I am going to help you with that. So autosome linked dominant trait and X linked dominant trait. So there is a difference in how do they appear. So X linked dominant and autosome linked dominants. Let's see, my dear children. So when it is autosome linked dominant trait, obviously one parent should definitely have this particular disease. It is a dominant trait, right? So obviously one parent should have it, then only the next generation will be having it. So you want with dominant traits, one thing very common to all dominant trait is that it will be found in every single generation. It would be found in every single generation. So there is no skipping of generation. You are finding it in every, this lady is diseased, the lady's son and daughter is diseased, again their daughter is diseased, their daughter and son is diseased. So every generation you see it. So that is one very important example, okay, or basically a tip that you can remember. You can find disease in every generation. Now how do you identify whether this particular disease is autosome linked or it is sex chromosome linked, that is X chromosome linked? That is also really easy. Okay, how do you, so this is diseased, okay, so don't think this is not diseased, this is diseased. Now over here, if you see, the mother is diseased, here the girl child and the boy child is diseased. Now again here the girl child is diseased, here also the girl child and the boy child is diseased. When the boy child marries, okay, it is going to affect the girl child and the boy child as well. But over here if you see, if there is a father, okay, if there is a father who is having the disease, he will be passing on the disease only to the daughters, no one else. So over here you see, the father is diseased, he is passing on to the disease just to the daughters, not to the sons. Why? Because it is what? X-linked disease, my dear children. So probably, I'll tell you, female over here is normal, suppose. The male is affected. The male is affected and this male has the disease. Okay, I am saying he has the disease, which is why this particular allele is there. And this guy is affected, so this is the parent and this is the female parent that you are having. Now when you are going to make a cross, you get this and this gamete, and here you get XH and Y gamete. Now when you are going to fuse it, let's try to fuse this and this. When you fuse, you get, oh my god, where should I should be doing it somewhere here, sorry. So X and X is there, and then what do you have, or should I do it here? I'm so sorry children, sometimes I mess around like this, I'm so sorry, but this is all for you to understand things. So X and X is there, these are the gametes made by the mother, and XH and Y is there, right? So this is a gamete and this is a gamete. This is a dominant trait, S-linked dominant trait. So if at all any one chromosome is affected, that means that person will be having disease. So over here both the X's are not having the disease, so it's okay. This female is fine. But here this guy has this, so she is diseased. Okay. Now the gametes when it fuse, this and this when it fuse, you get X and XH. This is a female, XX is for female, and she will be diseased because there is one X which is affected, and this is a dominant trait. So only one is needed for getting affected. Okay, now let's fuse this and this. So it is XY, my dear children. This guy is safe. This is a guy and he is safe. What about this? Again, a female who is going to get affected. So anyways, whatever you do, when it is X-linked dominant trait, the father will be giving the disease to both the daughters. The daughters are going to be affected anyhow. So you see that over here. Here there is nothing like that. Okay, both the sexes can be affected, but over here, if the male is affected, my dear children, the female also will be affected. The daughters will be affected in the case of X-linked dominant traits. Okay. So dominant trait, how do you identify? It is found in every generation. Now you have to look for whether the father is going to give the disease to the daughter and the son both, or only to the daughters. If it is passing down to only the daughters, that means it is X-linked disease. Okay, I hope I was able to explain you also. Now the next thing that you have is autosome linked recessive trait and X-linked recessive trait. Again, recessive trait, how you can identify is that basically there will be organisms, diseased people will be skipping the generation. For example, the parents might not be diseased. The parents might not be diseased. Okay, they might be carriers. But the children who are there, they might be affected. For example, over here if you see, she is a carrier. Whenever you see a small dot, that means that they are a carrier. So they don't show the disease. But the son who is there, he is affected. The son who is there, he is definitely affected. Okay. Now over here if you see, this is autosome linked recessive disorder. So the son, the husband and the wife, both of them, they are carriers, my dear children, and that's when the child will be having it, the girl child is having it, and the boy child again becomes a carrier over here. Okay. So it is a recessive disorder. So there should be recessive traits coming together. Okay. Now going ahead with the next one that we are having over here is this. Chalo. So this is what? Autosome linked recessive trait and X linked recessive trait. How do you identify between them? First thing, recessive traits is easy to understand because there the diseases skip the generation. For example, one generation might have the disease, the other generation might not have. So usually the parents might not be having the disease, but the daughter and the sons, they might be having it. So you have to see, they skip the generation. Recessive disorders, they skip the generation. So over here, the dots that you see, that means they are carriers. They do not have this disease. They are just carrying it. They can pass it on to other organism, okay, or their child basically. So how do you differentiate between this and this is that you see the boys and the girls, the male and the female, both of them, they can be carriers over here in this particular case. But when you talk about X-linked recessive disorder, my dear children, here only the girls can be carriers. Why? Because it is X-linked recessive disorder. Recessive, so it should be, for example, if the allele is affected, will this person be affected? No, she is just going to be a carrier. Why? Because this particular disease will be shown only during recessive condition. That means both of them should be having it. Here there is one to dominate one, there is one normal X chromosome to dominate this one, which is why it won't be able to show its effect. Only in this condition it will be able to show its effect. Okay. Now in the case of males, suppose one of them is affected and this is one of the allele is affected and there is Y chromosome. Is there anything to dominate over it? No. There is no one to dominate this particular guy. So this particular guy anyway will be expressed. So hence all the males that you see will be affected. If the males, they have this allele, this affected allele, they will be, this particular guy will be affected. But in the case of females, since there is one more extra X chromosome that we have, it will be able to overshadow the effect of that. Hence over here, it's not necessary that always the females will be like this. The females can be like this also, but in that case they are affected. But the females can be like this also, and the simplest thing is that here the carriers can only be females, no one else. But in the case of autosome disease, it can be both male and female who can be the carriers. Here both of them can, here only one can be the carrier because it is what? X-linked recessive trait, my dear children, and we have two X chromosomes. So one can be normal and hence we can be the carriers. Cool? Done? Fine? Chalo. Next, Y-linked inheritance. This is the most easiest one. In Y-linked, only the males are going to get affected, no females are going to get affected. If you see the square is for males and it is darkened, that means they are affected. So over here, generation after generation, the males are getting affected, which is why you call this as Y-linked trait. So for all of them, you have to give probably explanation. How many points? 2 points you have to give, one for each. Okay, 1 mark for each. So basically do not miss out those points. You have to give an example of the disease which is there and you can identify it properly and write it properly. Okay, so write point by point there. Identify the disease first. Identification is equals to this. Okay, this is the disease. Fine? Cool. Now question number 4 that we are having over here, my dear children, is identification and commenting on the things that are given below. Either of them will be given to you. Okay, it can be in the form of slides or in the pictures. TS of testis, TS of ovary, that is transverse section of testis, transverse section like this. Okay, and vertical section, that means like this. Fine. Now, okay, VS of ovary showing female gametophyte, VS of blastula, and comment. If you, for example, you are identifying any one of them, you have to give comments. Any 2 points you have to give, my dear children. Okay, you have to give 2 points for 1 marks for each point. So, how do they look exactly? So, exact picture I do not have, but yeah, it might be something like this. So, this is the TS of the testis. TS of the testis. So, this is the testis, suppose. I am cutting like this and I am observing it like this. Okay, I am observing it like this. That is what you mean by transverse section. So, what do you see? What do you see? So, they have seminiferous tubules, my dear children. Inside the seminiferous tubules, there will be sperm formation taking place. So, this is the seminiferous tubules. Now, seminiferous tubules, my dear children, okay, in between all the seminiferous tubules that you are having, there will be certain spaces. What do you call the space? Interstitial space, where you have what? The Leydig cells. Where you have what? The Leydig cells. So, you will be finding circular things like this where sperms might be floating. This is the seminiferous tubules, and in between what you have? The interstitial space where the Leydig cells are there. Okay, so this is the TS of testis, where you have got these many circles over there. Now, when I talk about this, this is the ovary. Okay, this is what? The ovary that you are having. Okay, of human beings, TS of the ovary. So, it will look something like this, where the Graafian follicles and all the follicles will be mentioned. Okay, we will be seeing there will be smaller primary follicle, secondary follicle, Graafian follicles, and then you will see that one follicle is degenerating, that is forming the corpus luteum over there, and things like that. Okay, again, it can be a picture, it can be a slide or something like that. The pictures can be different in your school, so be careful about that. I hope your teachers have shown you. Okay, but you should be knowing the points for each. You should be knowing the points for each of them. Now, then we have the VS, okay, the vertical section of an ovule. Now, the vertical section of an ovule can be given like this or can be given like this directly, where the female gametophyte is seen. Okay, you will be having 3 cells over here. These 2 cells are the synergids having the filiform apparatus. This is the egg cell. You will be having 3 antipodal cells over here, and you will be having 2 polar nuclei over here. This is what you mean by the embryo sac, the female gametophyte. So, VS of an ovule. Inside the ovule, what do you have? You have an embryo sac. The same thing is being mentioned over here. This is the VS of the ovule. Okay, this is the whole VS of the ovule, and this is what? The embryo sac, my dear children. This is the embryo sac. Cool. So, you might be given a picture like this or a picture like this. You have to identify. Picture as in I am talking about slides. Okay, do not get me picture. Sometimes they can give you a picture also, but mostly you do not get the picture. You get slides, and you have to understand what is it. Now, when there is a ball-like thing, and in between the ball-like thing, there are so many other cells also. That means we are talking about blastula. Okay, the VS of blastula, vertical section of blastula that you are having. This is a stage of embryo development where you have the outermost cells arranged like this, and what do you call them? You call them trophoblast layer. And inside that there will be certain cells arranged like this. What do you call them? You call them inner cell mass. Okay, and there will be lots of content over here. This is known as blastocele, my dear children. This is a fluid filled cavity that you have that you call as blastocele. Okay, so make sure you are able to identify all this thing. You are able to write something about each of them. 2 points. Fine. So this will be pakka from any of these. So you have to be sure about any of them. Now. Question number 5. So identification of any one among the given below, that is about the disease causing organisms. So you have Ascaris lumbricoides, which causes ascariasis. Okay, you have Antamoeba histolytica, which causes amoebic dysentery. You have Plasmodium vivax, which causes malaria. You have ringworm fungus, that is Trichophyton rubrum. So you have to, they won't give you the name, you have to look at the picture and you have to identify it. Again, the slide, the picture, whatever is it, you have to identify and make sure, my dear children, the genus name, it is starting from capital letter, the species name, it's a small letter. Okay, it is a small letter, and you have to write it completely like this without any spelling mistake. Your spelling should be correct. So how do you make your spelling correct? There are only 4 things. Write it repeatedly and learn it. Okay, and you have to give what comments to each of them. Any 2 points you have to give. For example, Ascaris lumbricoid you got. Okay, you went ahead, you identify it, and now you have to write 2 points about it. For 1 point each, for 1 point you will get half marks. Okay, for each of the points you get half marks. So don't go write that blunder. Know what you are writing over there. Analyze it now only. Okay, when I am talking to you, just know Ascaris, it causes ascariasis. Okay, it is going to block the intestinal passage and things like that. So what does it cause? Okay, what are the symptoms of the disease? How is it passed? Things like that you should be knowing already, so that you don't juggle up there. Okay, so yes. Now the last thing that we have over here is question number 6, my dear children. So again, you have to identify any one of the things that is given below. Either it can be controlled pollination part, or it can be homologous and analogous organs of plants as well as animals. Can be plants or animals. For example, if you're talking about homologous organs of plants, then you have let's say tendrils of cucurbita and thorn of bougainvillea. So thorn and tendril both are arising from the eggshell bud, okay, but they are developing into different things, okay. That is thorn and one is tendril, right? So they are having a common common ancestor, my dear children, and they got habituated differently, which is why they moved away differently, forming different organisms or organs, okay, which you call as homologous organs. So this is an example of divergent evolution. So you should know that homologous organs are seen in divergent evolution, analogous organs are seen in convergent evolution. Now wherever the functions become different, just understand it's homologous organs. Whenever the functions become different, just understand it is homologous. Whenever the functions are same, just understand it is analogous. So in analogous organs, even though the functions are same, they are not from common ancestors, okay. For example, wings of birds and wings of butterfly. So butterfly wings is just what? Just a small thing that you see. Wings of birds will be having fold, they will be having what? Bones over there, things like that, flesh, right? So it is totally different, but both of things are being used for flying. So it's because they are in a common habitat, that is why they have this. But do they have a common ancestor? No. So they are showing convergent evolution, and these organs are known as analogous organs. Okay, so for plants and animals, you should be knowing about examples which are already given to you by your teacher. You should just go through it. Now, emasculation, tagging, bagging and everything, you should know that also. Emasculation is basically when they are going to show you a plant, the scissors on it. That means basically you are removing the male part over there. You are going to remove the male part over there. That is what you mean by emasculation. Okay, for example, you have a flower like this. Okay, with a stigma like this and also stamen. So you do not want the pollen grains to land over here. So what do? You cut them. You cut it so that this particular female reproductive part is left like that, so that self pollination does not happen. Okay, that is what you mean by emasculation. Now what do you mean by bagging? Bagging is when I cover this up so that no pollen grains, basically other pollen grains come on it. Okay, that is what you mean by bagging. And I wait for the stigma to become receptive. When it becomes receptive, I will put the pollen grains that I want to put over here. Okay, so emasculation, bagging, either they can give you a picture, okay, or they can put a pot and just bag the flower. Just understand it's bagging. Okay, it's part of artificial hybridization. Emasculation and bagging is a part of artificial hybridization. Now once you have identified what is there, identification gives you 1 mark, and commenting will be giving you half marks each for each point. So basically 1 mark at the end. 1 plus 1, 2 marks. Okay, so that is all about the experiments. I know you might be tired listening to me, but yes, it is how it is. So yes, my dear children, now last but not the least, records are necessary for breaking the records. So if you want to break the records and score really good marks, my dear children, you have to maintain a proper record. Okay, so according to them, okay, they have given number of experiments to be performed and recorded, and how many marks you will get accordingly. So if someone has done less than 75% of exercises, you're not eligible only. Okay, now 75 to 79% of exercises, 3 marks you will get. So why not just take the 6 marks that you can easily get? Okay, so more than 90% of exercises if you have done, my dear children, so you get 6 marks. So please make sure you're having a proper practical record. Take it very seriously. If you have not completed it, please complete it. Okay, because you have your exams and you do not have to lose 6 marks of it. Children, don't think that you're losing marks over here. That 30 marks, no, it's not a problem. Basically, these 30 marks are going to come in your cumulative marks, right? 30 plus 70 is equals to 100. So your marks are going to get hampered there also. So why not take it really seriously and do something about it? Okay, so please maintain your records, and when you're going for Viva, make sure you're knowing about each and every detail about the experiments that you are doing, you're performing. So it's not like, as I said, out of the 4 experiments, you can get any one experiment, right? So you should be knowing about all 4 of them. But eventually they will ask the one that you're getting mostly, but you do not know what you're going to get. So it's better to just go through it. Why are we using the stains? Why do we treat it? Why do you treat it with alcohol? Why do you heat it? All these things are really, really important. What do you have to make sure of? Do you see in lower magnification first? You say yes. Okay, what is the lens over here, the microscope? The one that we have near the eyes, it is known as ocular lens. The one that is near the specimen is known as objective lens. Okay, so you should be near the object, it is objective lens. So all the small details are really necessary for each of the things. And whatever you are being asked over here, that means there are a lot of theory behind it also. The annulus organs and things like that I just said. There are so many examples, you should be knowing about it. Okay, so that's all. So just go through the NCERT really well before going to exam. Okay, so whatever theory you're not able to understand, just go read it. You will be able to understand things better. And we are here to help you. So do let me know if you need my help. Okay, so that's all. If you're ready to score 30 on 30 now, do let me know in the comment section. I will be so happy to see your comments. Okay, because I have put so much hard work into finding the practicals and how you should be doing it, because it's long time since even I didn't do it. So yes. Cool. So that's all. Yeah, so that's all, my dear children. And if you are liking our video, do not forget to like, share and subscribe to our cute little channel, that is Diksha Karnataka, and do not forget to show your love with the help of comments. Bye bye, take care.